Até conseguimos fazer um terceiro dia, diz lá.
Conseguimos. Não, claro, com tantos temas tão interessantes, conseguimos fazer um terceiro e um quarto. Talvez fica a ideia para o próximo ano, Catarina.
Fazemos uma jornada de 3 ou 4 dias, uma jornada contínua e assim. Muito obrigada desde já à presença do Sr. Presidente do Governo Regional. Muito obrigada à Sra. Presidente da Câmara por estarem connosco hoje. Hoje somos nós os anfitriões. Desde já vamos iniciar a última parte.
Sim, vamos então dar início à conferência do Sr. Professor Craig de Mello, que é o nosso convidado de honra, que começa beijamente conhecido o Prémio Nobel de Fisiologia ou Medicina de 2006 e que será introduzido pela Sra. Investigadora Luísa Mota Vieira, coordenadora da Unidade de Genética e Patologia Molecular do Hospital do Divino Espírito Santo de Ponta Delgada. Pedi então à Sra. Investigadora o favor de subir a palco.
So, good afternoon. Thank you all for physiology and medicine in 2006. So we are very happy that you accepted the invitation of the International Hospital of the Azores, particularly to be here today with us in the island of San Miguel, the island of your paternal ancestors. So I think it is your fourth visit to San Miguel since the last one was in July of 2018. It is a great honor and a big challenge for me to introduce you to the audience since you have an extraordinary career. Your biomedical research and discoveries are opening new avenues in the treatment of disease, for example, by using smart drugs. This is a topic of extreme importance and relevance for patients and physicians. Also, I would like to give a special thanks to the Dr. Isabel Casio, who is the clinical director of the International Hospital of the Azores, for inviting me to introduce Professor Craig Mello. So now I will speak in Portuguese about your Azorean ancestry and family, your journey as a scientific novel, your awards and honors, and the applications of the RNA interference as a new therapeutic area based on RNA. So, portanto, vou falar agora em português. Craig Mello nasceu nos Estados Unidos, em New Haven, é 18 de outubro de 1960, portanto fez anos muito recentemente, portanto os nossos parabéns atrasados. É o terceiro filho de uma fratria de 4 irmãos, sendo filho de James Mello e de... Eu acho que foi por aqui. Que era paleontologista e há aqui um destaque, era doutorado e doutorou-se pela Universidade de Yale em 1962 e a sua mãe, Sally Cameron Mello, fez das artes a sua profissão. Portanto, viveu e cresceu num meio particularmente rico, em que os pais fizeram da educação, do conhecimento e da descoberta uns pilares nessa família. Portanto, é neto, pelo lado do pai de Frank Mello, e Frank Mello é descendente direto de açorianos, embora já tenha nascido nos Estados Unidos, em Rhode Island. Portanto, Craig Mello é então bisneto por lado paterno, de Eugênio de Melo e de Maria de Glória Baracua, e este casal que emigra da freguesia da Maia da Ribeira Grande, para os Estados Unidos, levando consigo 5 filhos. E esta imigração ocorreu no início do século passado, mais precisamente em 1907. Este casal, quando chega aos Estados Unidos, tem 2 filhos. E esses tem mais 2 filhos. E é o 7º filho que vem a ser o avô do Craig Mello, e que este avô, que é por quem Craig Mello tem uma grande estima e consideração. Ele refere isso com alguma frequência nas suas entrevistas que deu até na atribuição do Craig Mello, do Prémio Nobel. Craig Mello tem 2 filhas, eu não consegui arranjar fotografias mais recentes, elas hoje são jovens universitárias, mas são 2 bonitas raparigas. Pronto. Relativamente ao percurso académico do professor Craig Mello, ele inicia o seu percurso académico em 1982, licenciando-se em Bioquímica e Biologia Molecular pela Universidade de Brown. Depois desloca-se, muda-se para a Universidade do Colorado, onde faz um curso de pós-graduação em Biologia Celular e Molecular e do Desenvolvimento. E este curso vai ser determinante no percurso do professor Craig Mello. Neste curso, ele encontra um colega mais velho, um colega este que é convidado a montar um laboratório em Cambridge, na Universidade de Harvard, e convida Craig Mello a juntar-se a esse projeto. E o professor Craig Mello aceita este desafio e vem com o colega para Cambridge. Em Harvard University, faz o seu doutoramento, e o seu doutoramento ele utiliza como modelo animal o nemátodo, que é o verme da terra, que é o nome científico C. elegans, e utilizando este modelo animal, ele colabora, os seus estudos levam-no a que ele colabore com um colega, Andrew Fire, que trabalhava na altura em Baltimore. E esta ligação, esta colaboração vai ser determinante nas descobertas que se vão seguir. Mais tarde, uma vez terminado o doutoramento, o professor Craig Mello muda-se para Seattle, onde realiza o seu pós-doutoramento no Instituto do Cancro. E, findo este pós-doutoramento, obtém uma posição na Faculdade de Medicina da Universidade de Massachusetts, onde hoje é professor e docente da disciplina de Medicina Molecular, tendo o título de professor galardoado. Foi o melhor termo que eu encontrei para a tradução de Distinguished Professor. Fui eu consultar o site, a página oficial do professor Craig Mello na Universidade de Massachusetts, na Faculdade de Medicina, e realmente vemos o número de publicações importantes que ele tem, que está mostrado desde 1996, em várias áreas da ciência, que realmente esses artigos todos publicados nas revistas mais prestigiadas, como a Nature, a Cell, enfim, de grande renome. E aqui as publicações, e ontem falámos um bocadinho sobre isso, as publicações são o meio de excelência para a divulgação do conhecimento científico, no qual as sociedades científicas também têm o seu papel. Com este percurso académico e de formação, e com os trabalhos e os artigos que o professor Craig Mello foi publicando, várias instituições foram reconhecendo ao longo do tempo o trabalho, atribuindo prémios e distinções. E começamos então em 2000 com o primeiro prémio, é nomeado investigador médico do Instituto, que é muito de grande prestígio. Em 2003 recebe 2 prémios, 2004 mais 2, 2005 mais 2, 2006 recebe o primeiro para este instituto, que é alemão, e ficou na shortlist, não é, dos candidatos a Nobel. E, por conseguinte, passado uns meses, em novembro, ele realmente é nomeado em exequo com o seu colega Andrew Fire com o Prémio Nobel de Fisiologia e de Medicina em 2006. O trabalho que levou à atribuição, ao reconhecimento do Prémio Nobel, baseou-se na descoberta do RNA de interferência. RNA é um mecanismo esse que permite o silenciamento dos genes, eu vou explicar isso de uma maneira muito simples, silenciamento dos genes esse que também participa no controle da informação celular que ocorre no interior da célula. É um mecanismo que está lá sempre para proteger e garantir que a célula está a funcionar corretamente. E está aqui o artigo que realmente foi reconhecido. Pronto, aqui só para relembrar e para nos futuros mais novos, aqui a atribuição quando recebe o prémio pelo rei Carlos Gustavo da Suécia, aqui o diploma e a medalha, e todos os nobres, durante aquela semana única que alguns têm o privilégio e são raros de ter, é o que o professor Craig Mello dar uma lecture no Instituto Nobel na Suécia. E aqui está os premiados do ano de 2006. Pode passar? A seguir à atribuição do Prémio Nobel, muitas distinções e um grande reconhecimento houve por muitas universidades e por algumas sociedades científicas. Destaquei 2. O professor Craig Mello tornou-se membro da Academia Americana das Artes e de Ciências, na área de especialidade de Ciências Biológicas, e a Universidade dos Açores, em 2012, em janeiro de 2012, atribuiu o grau de doutor honoris causa numa cerimónia que foi muito bonita. Aqui tem a fotografia que está no site oficial da Universidade. Pronto, e para terminar, gostaria de referir o seguinte. Desde que o professor Craig Mello descobriu, e André Fire, o mecanismo de RNA de interferência, até aos nossos dias, passaram-se mais ou menos 20 anos. E durante estes 20 anos houve um grande desenvolvimento e uma nova abordagem terapêutica baseada nos RNAs. E esta imagem, que é recente de julho de 2022, mostra o RNA como arma terapêutica, não só utilizando os mecanismos do RNA de interferência, do silenciamento génico que eu falei, uma vez que já há ensaios clínicos em curso, bem como, também olhando aqui para a esquerda do gráfico, as vacinas de mRNA, de RNA mensageiro, que foram tão úteis no combate à pandemia e, se me permite, a par dos testes, do qual tive os testes de detecção do vírus. Portanto, foram duas várias, mas cá está, em 20 anos, abriu-se um mundo destes novas drogas, novos fármacos, no combate às várias doenças. Eu digo no combate, mais no tratamento das doenças e não na cura. Às vezes pensamos que todos os medicamentos curam, mas a maior parte das vezes os médicos tratam as doenças. E, portanto, sem mais demoras, vou passar então a palavra ao professor Craig Mello. Please. So it's a great honor to this meeting to attend your conference. Please. Thank you.
Thank you. Thank you. Thank you for that very kind introduction, and it's wonderful to be here. I hope that the translator is available to you. She's been doing a wonderful job for me. So I think you can access it. It'd be wonderful if you could. I'm going to use this mic. And yeah, so it's really exciting and wonderful to be here. It's wonderful to get to meet the president and to meet and to learn, my colleagues here at the hospital and at the university, and of course my family from HIA. It's wonderful to see you all again and to visit these beautiful islands. When I flew in the airplane and I could see those green hills, I just felt like I was coming home, which is nice. And I thought of my great-grandparents seeing those green hills disappearing on the horizon, never to see them again in their life. Because they couldn't afford to come back. So kind of got a little emotional thinking about that. So I'm very fortunate. One of the things I always say is that I was incredibly lucky to have such wonderful role models, not only from my parents, who were mentioned in the introduction, but my grandparents, who never had the chance to go to even finish high school. None of my grandparents. On my mom's side, too, they were also working class, hardworking people who never got to finish high school. And what was wonderful for me as a scientist was that I could appreciate their sacrifices and the hard work that they did that made it possible for me to have the chances I had. And I just really, it made me respect the common folks who make up most of our society, who don't get the chances that a lot of us get, most of us in this room, to go on to college, for example, or even to finish high school, but they have to work to help their family. There's a lot of really smart people out there who just haven't had the chances that we've had. So again, it's wonderful to be back, and it's wonderful to have to feel these connections. So I'm going to tell you two quick stories. I'm not going to give you a lot of details, but I want to convey a couple of messages. First of all, that organisms thrive on information. Information is life. That's what makes us work. And us, all living things, have been living in the information age for billions of years. Billions. That's a really, really long time. The Earth is very old. I'll talk about that a little bit more in a moment. But when I was a kid in high school, 1977, I read in the newspaper that the human gene for insulin had been put into bacteria, and that bacteria could make the human insulin protein. And everyone was excited about that, and I was too. I thought that was wonderful, because think of the medicines that you could make, insulin, for example, but many other proteins that could be helpful to people by using bacteria. Would be really quick and efficient. But what really surprised me about this article was that bacteria could actually read the human genetic code. Humans and bacteria speak the same language. And we all know languages can change very rapidly. I wish I could speak Portuguese. It would make this much more enjoyable for all of you. However, in the history of life, all of the organisms alive today use the same language. How could that be? That got me fascinated, made me want to study biology. I wanted to become a biologist. That was the time, that was the day for me when I read that. I wanted to figure out why do we speak the same language? Why do all organisms speak the same language? And this is the animal that I ended up working on. It's called C. elegans. And I like to say that this is the real Nobel Prize winner. It's won 6 gold medals, 5 in medicine and 1 in chemistry. And the reason it's so medically relevant is that humans and this little worm shared 3 billion years of common ancestry. Since the origin of life on the planet to about 800 million years ago, we were the same organism, we and this little worm. This is what evolution tells us. It's what our DNA sequences tell us. If you sequence this little animal, it's very similar to a human. If you look at its muscle, the muscle is very similar to a human. Its nervous system, very similar to a human. They are very, very much like us. And importantly, they handle information the same way we do. So when I was a graduate student, I started working on this little animal to find out how it handles its information. How does it incorporate new information? I was using a tiny needle to inject information. This is a gonad that's right here. If you put new information into the gonad, we call that DNA transformation. You can put information into an organism. The organism can use that information, express the genes that you put in. Now we would call it, in the case of a human, we would call it gene therapy. It's a way of putting information back into the animal. And Andy Fire and I were both working on this really tricky, hard problem. Had never been done in any animal. And of course, these little tiny animals were great to use as a model system, as a first step in trying to understand how to put genetic information back into an animal. Many of you know that it's very easy to break something. In genetics, when you break something, you use mutations to break something, typically. And until we started working on this, at the time when we started working on this, there had never been a case where you could put a corrected version back in and repair what you broke. So that's sort of the tool that we were trying to make. And anyway, I won't go into all the details, but I do want to talk more about, can we have the lights down a little bit? This is a kind of a dim slide. I want to talk more about information. How do organisms handle information? Well, this is the ovary of this little tiny worm, and you can see these are the eggs that are being made. And I just want to point out a couple of things. The DNA is inside these nuclei. That's where the DNA is stored. But there's a lot of other information in this cell as well. These green things that are labeled here in green, these are mitochondria, which are they're like bacteria that are now a part of our cells. So the ancient eukaryote, the cell that gave rise to both the worm and the human, had these bacteria living inside of it, believe it or not. And a symbiosis developed where the information that makes the bacteria moved into the nucleus along with the... I know this is going to be hard for you to understand. I apologize. I really shouldn't even be showing this slide because it would take me an hour to explain it adequately. But let me just say this. Information is in here in the form of DNA. Information is also here in the cytoplasm. There's DNA inside of each of these little green things, which are like bacteria. But this organism has become a melded organism. The ancient origin of these green things were separate cells, like bacteria that infected the primordial cell. And this is billions of years ago. This symbiosis, this movement of information out of the green stuff into the nucleus has happened over hundreds of millions of years. What I'm trying to say here is information flows not just from your parent to the offspring, but information flows horizontally from one organism to another. We've all experienced this recently with COVID. COVID is information gone viral. It's information that moves from one organism to another, okay? So the concept here is really fascinating, because what organisms are telling us is that information flows between different species. Across evolutionary history, information continuously is flowing from plants to animals, from fungi to animals, from animals to fungi, to bacteria. Information is moving all over the place, not just coming from your parents, but also coming from your environment. You are constantly exposed to new information. Anyway, this is just another way of illustrating this. This is the history of life on the planet. The origin of life, about 3.8 billion years ago, is thought to have occurred by essentially molecular evolution. Organisms, I feel bad for the interpreter trying to interpret all of this too, if you guys are listening that way. But basically, information that could self-replicate got together gradually over early evolution, we think. This is based on theory. And the DNA genetic code arose. And then this was passed to the various types of organisms that were alive, mostly microbial life, bacteria, archaea, and these things, cyanobacteria. These organisms here became photosynthetic, gave rise to algae and plants later on. But if you look at what happened in the history of life, information flows together. Multiple organisms share information. Again, bacteria and archaea undergo this mitochondrial symbiosis. I showed you the green signal inside the cell. Those were mitochondria. They fused, organisms fused together, shared information, and gave rise to, all the different organisms here, plants, fungi, and animals. All had a common ancestor. Again, fusion between the cyanobacteria and these early eukaryotic cells gave rise to plants and algae. And when the fusions between living things occur, they share their genetic information. It's like giving rise to a new child. You have the mother and the father sharing genetic information. In these events, you had two independently evolved organisms blending together and sharing information. This happened repeatedly in the history of life. Our genome is full of bacterial DNA. We have more than 1,000 essential genes that you could not live without that came from bacteria. We are bacteria, basically. We are. We're closely related to them. We're descended from them. But more importantly, we share information all the time, not just from our parents to our offspring, but on an evolutionary time scale, on the scale of millions of years, information flows from other organisms into humans, into our human lineage, and from our lineage into others. This is why all organisms on the planet use the same language. because if you can't speak the language, you're at a huge disadvantage because you can't acquire useful information from other sources unless you speak the language. You have to be able to speak the language. There's one language for the whole planet. Every single organism speaks that language. It's so cool, so amazing. After billions of years of evolution, every organism on the planet speaks the same language, and they share information constantly. There's no such thing as a non-GMO. There's no such thing. Everything is genetically modified constantly. That's how life works. And so anyway, this is just the timeline, the history of life. 3.8 billion years ago, the first cells. 2.7 billion years ago, photosynthesis. That's a long, long time and a very sophisticated mechanism. The cyanobacteria made oxygen, put it into the atmosphere, and they froze the planet. For 100 million years, the Earth was frozen to the equator. There was no land. They almost wiped out all life on Earth by messing with the atmosphere. Can you imagine an organism stupid enough to mess with its own atmosphere? That's what the cyanobacteria did. So here, there were a couple more of these. This blue line, that's a global glaciation event called a snowball Earth. There were two more of these at least. One about 100 million years below the Cambrian was the most recent one. And I want to draw your attention to this area. This is the area that I call the era of super sophisticated tiny organisms. These are animals like the worm that I worked on that are microscopic. They live in the soil, in the ocean, and at this time, all life was in the ocean pretty much. There were no land animals or plants. And then something amazing happened called the Cambrian explosion. Life blossomed onto Earth. It got big. We had fossils. We had cool things like big fish and crabs and all these things. But they were just bigger versions of these tiny animals. All of the major phyla, all the different kinds of animals alive today existed in the oceans back here. But none of them were on land until after this event. In fact, until a few 100 million years after this event. And let's see what else is on here. Oh yeah. So this is the dinosaur extinction. So the era of mammals is right here. There's a dotted line you can't see here, but it's about the same thickness as that line. That's the distance between humans and other primates like chimpanzee. It's also about the age of the Azores right here, a few million years. So the history of life is really long. That's the message. Organisms are incredibly sophisticated. They have amazing information handling ability. They can access information from other organisms. They can handle it safely. And what are some of the things they do? So they're great at assimilating information, but they use silencing when they get new information. They're really smart about it. Humans are not so smart about how we handle information. We are messing up big time when it comes to handling information. Organisms are super careful about information. They don't believe it at first. They make sure it's safe first, and the way they do that is they silence it. And I'll be telling you about this silencing machinery that they use, which we discovered completely by accident when we were trying to put information back into the animal. That's when we discovered that they can silence information very efficiently. And they can keep it silent without destroying it. So essentially what they do is they gather all the information, and then they figure out which parts of it are safe and which parts they can use. And the way they do that is by keeping it silent for the most part, but letting some progeny, some of the progeny that are born, and there are millions of them, express a gene, for example, and that if the gene is helpful then the organism will benefit, and there'll be selection, and that organism will have more progeny, et cetera. It's the whole Darwinian thing, but basically they're doing it by carefully handling information. There's things, in fact, half the human genome is viral DNA, and things we call transposons. They're also called selfish DNA because they copy themselves and they hop around. And you've probably heard of junk DNA. Most of our genome doesn't encode anything obvious. It looks like junk. Transposons are often considered junk, but they are jumping around and doing their own thing, and in the process, they're rearranging information. They're essentially helping the organism digest it. So you bring in new information. Let's say you acquire an infection, or you acquire the entire genome of a microbe. It gets inserted into your own genome in your germline and the cells that give rise to your egg and your sperm. Those cells are the ones that are going to pass the information on, but you can handle it safely because you silence it. And then the transposons rearrange it. They jump it around, they move it around, they mix it up, they mutate it, and they essentially digest the information. They allow the diversification of the information into new programming. So germlines digest information and rewire it into new circuits and programs, and that's how we get evolution. Here's what the Earth looked like 640 million years ago, courtesy of the NSF. Basically, we can infer this. There was no land, and so there were no land animals, but the organisms that were alive in the ocean were just as sophisticated as we are. They had already reached incredible level of evolution in their ability to handle information because they're incredibly ancient. One of the things I love about science is that it's so weird, you can't make it up. You know, we're all human beings. We're all here, and we don't know why. I call that the human condition. You just show up one day, and you're alive, and you have to come up with some explanation, like, where did I come from? And the real, if you study the world, the cool thing about it is as you study the world, it becomes more weird, not less. More weird. I mean, just look at the Big Bang and quantum mechanics and all of these weird things that people say are sort of the underlying basis of our history and our natural history. It's cool. It's really cool. This is what the Earth looked like, and in the oceans of this planet, there were organisms just as sophisticated as we are. They had all of the major innovations that we have, all of the tools that we have. No, they weren't big and didn't have a brain like ours. I wouldn't call humans very smart, though, either, you know. Anyway, what I like to say is that the genetics is so simple. It's such a simple language. Everybody should know this. There's only 4 letters in the alphabet. Here they are. The cool thing about them is that they can pair. The T's and the A's pair, the G's and the C's pair, and they can form this beautiful polymer called your DNA, where the information in this strand predicts the information that's in this strand, and then you get that beautiful staircase helix that Watson and Crick discovered. Information, so simple, really, really simple. A polymer that replicates, that drives, underlies the genetics. And there's only 20 words. We call these words amino acids. The order of the nucleotides, the DNA sequence, determines which amino acid gets put into the protein. The amino acids are then formed into a chain. And instead of getting one structure, like that beautiful helix, you can now make snowflakes, because each of these little amino acids, each of the 20 words are different. You get these amazing structures. For example, you can get the spike protein of COVID-19. by making an mRNA that has a sequence like this, very simple. The sequence of this DNA strand determines the sequence of what's called an mRNA that serves as an intermediate. It's basically just one of these strands of information after some processing. These get incorporated according to a very simple genetic code defined by reading three nucleotides at a time. You assemble another polymer, and you can see how the protein is arranged by making a polymer out of these little 20 building blocks. It's so simple. That's how you make the mRNA vaccine. You can make anything, you can make any protein, basically, by just transcribing an RNA from a DNA strand. You can make an RNA strand that gets translated by these little things called ribosomes into another polymer, but this one has much more complexity because instead of just 4 building blocks, it has 20. And the 20 building blocks in protein have much more diverse chemistry. So this little movie just illustrates the point of what RNA and DNA are. I'm just going to let the soundtrack play on this.
Every creature, and you know this from high school, is made from a recipe that comes from its DNA, spelled out in chemicals, A's and C's and T's and G's, inside the famous double helix. Every creature has its own DNA, different from mice, and then for whales, and for flowers, but to go from a chemical recipe, A's, G's, and T's, to a real creature that squeaks, or soars through the air, or turns gloriously pink, that requires RNA. RNA is the thing that turns you from a chemical code to a real, pulsing, living creature. RNA builds life. That's big.
Yeah, I love that. So RNA is the software, right? DNA is the hardware. RNA is the software. Your body is filled with information. That's what makes you work, and when information gets broken, that's what makes you sick. Information is what the virus was made out of. It infected us. It took over our cells. It used our cells to make more virus, and it was bad. And it could be much worse next time. We were lucky. This one didn't kill our kids, at least not as much. But it's information that lies at the base of it. So RNA is the program that's running. So if your neuron expresses different genes into RNA than your skin, and so your skin looks different from your neurons, and muscles are different also because they're running a different program, but they all have the same DNA. So it's really kind of cool. And of course, incredibly sophisticated. It's another little movie showing you, again, this is the ancient mechanisms part of the talk, and I'm almost done with it. But here's the DNA wrapped up inside the nucleus. There's tons of it in there, so it gets coiled up like that. And now you can see the polymerase getting on. This is called the RNA polymerase, and it copies the DNA, this little green blob. It copies the DNA to make the RNA. Here it goes. So it's reading the DNA sequence and it's making an mRNA. And in real life, this thing copies at a rate of 50 nucleotides per second. That's fast. This is obviously slowed down incredibly. So here's the RNA is getting processed to make the mature mRNA. Then it gets exported out of the nucleus. And once it's out in the cytoplasm, the code that's here in these letters is going to get read by this thing called a ribosome, which then incorporates those amino acids I was just telling you about, and then the protein starts to fold up, and now you've got your protein. And this is the mechanism that we discovered called RNA interference, and we injected a double-stranded RNA here. It gets processed by the cell and loaded onto this protein, shown in this movie as a big purple blob. I'll tell you a little bit more about this protein. This protein uses the information in this piece of RNA to find matching information and cleave it. And that process is also very rapid. In fact, this protein functions at the rate of 100 million contacts per second, 100 million. So it's incredibly efficient. It's a search engine. This is what it looks like. I told you proteins have these beautiful folded structures. This is a single polymer of amino acids, and it's holding onto a guide RNA here. I think the guide is in blue. Hold on, maybe it's red. Yeah, the guide is in red. So it's holding onto a guide RNA, and it's using the guide RNA to find matching information. This is the search engine of the cell. It's Google. Cells have a search engine. This is why the worm got a Nobel Prize, because we found the worm's search engine. We found it in the worm, in this little tiny worm. And what was cool was when we cloned the gene that encoded this protein, humans had it. Not only did humans have it, but humans were using it for the same thing that the worms were using it for guided search. Humans have guided search. Every single one of our cells uses guided search. We have a Google in every one of our cells. I told you it gets weird. It's really weird. It's so cool. But what's super cool about it too is when we discovered this in the worm, we realized we could make the queries ourselves in the laboratory. We could make a query, give it to the animal, and the animal would carry out the search for us. That means that we could make a therapy that would inactivate a gene that's causing disease simply by making a short RNA guide. Many of us in the room had the mRNA vaccine. That's a long RNA that encodes a protein. This protein uses the guide. It holds really tightly to the guide, and it uses the information in the guide to search the cell for information that matches the information in these 20 nucleotides. Just like you, if you want to use Google, will type to be or not to be, and you'll get Macbeth. You don't have to remember the whole play. You don't have to remember the poet or the playwright. Just remember one phrase, and you get the whole thing. Cells figured this out more than 1 billion years ago. Not only do humans have the same search engine protein, so do plants, so do fungi, and so do some bacteria. All living things use guided search. That's how information is handled. You could not use the internet if you didn't have guided search. It would be totally useless. Cells figured that out a long time ago. So now, with the discovery that there's a search engine inside of every cell, a lot of companies got interested in making drugs. And they started trying this right after the early clinical trials were only even before the Nobel Prize. And of course, maybe luckily for Andy and I, they hadn't failed yet in 2006. When we got the Nobel Prize, a lot of money was getting poured into making drugs for humans based on RNA interference by essentially making guides that were made out of RNA and then trying to give them to patients the same way we give the mRNA vaccine by encapsulating them in nanoparticles and delivering them into people, and it didn't work. Nothing worked. And this is just showing that, the amount of money that was going into this, and then Big Pharma pulled out. Basically, nothing was working. There was too many side effects, and it turned out that RNA was not a good drug. It needed to be modified. The chemistry needed to be modified, because normally when guide RNAs are made, they're made inside the cell. The cell makes the guide RNAs itself, of course. If you make them in the laboratory and give them to the patient, then the RNA molecule has to get into the cell. It has to go from the blood into the cells, and then, or from the cerebral spinal fluid into the brain, and it gets destroyed. So when a reporter asked me what I would have done differently after it had been right about here in 2016, they said, it's been 10 years, there are no drugs. What would you have done differently? I said, I would have hired more chemists, because that's what we were doing. We were hiring more chemists. At UMass, we established an RNA Therapeutics Institute, and at the time, it was a purely aspirational name. There were no RNA drugs at the time. There was no, in fact, I think a lot of people thought you couldn't make them, because we started right here when everything was falling. We started this RNA Therapeutics Institute, and we hired chemists who began to try to figure out where we could put stabilizing chemistry onto the RNA molecule so that it would be able to survive being delivered from the outside to the inside. And of course, this worked. It also worked inside industry, separately from the academic enterprise. In particular, it worked for liver delivery. There was a type of what we call a conjugate called GalNAc that could be attached to chemically stabilized RNA queries. We call them siRNAs. That's the lingo for the artificial guide RNAs that get loaded onto that search engine inside the cell. GalNAc delivers it to the liver. The first product was approved in 2018. This one just went through an amazing clinical trial, now available here, called Leqvio, lowers LDL by 50% over statins, and you need one injection sub-Q, meaning like an insulin injection into the subcutaneous fat, and your cholesterol will be lowered by more than your statins, if you're taking statins. You can pretty much eat anything you want, and you won't have high cholesterol with this therapy. Not available yet for everyone, but probably will be soon, because in the clinical trial of more than 4,000 patients, the only side effect was this mild skin rash in some individuals at the site of injection. But one dose lasted for 6 months. So you don't have to take a pill every day. And that was based on chemistry that was 10 years old. New chemistry means that you can probably get the dosing down to once a year. So it's like being immunized against heart disease. And you know how they found this target? The target of this drug was found from human genetics. People who don't get heart disease have mutations in this gene called PCSK9, and they're lucky because they basically never get hardening of the arteries. They never have I shouldn't say never, but they hardly ever have heart attacks. So these people became interesting because of their better than expected health outcomes, despite a bad diet, for example. And so they determined through genetics that the reason was that they had a mutation in this gene. This drug targets and lowers the activity of the gene that is mutated naturally in a subset of people who are resistant to heart disease. That was how that drug was discovered. And now you can now take this and have the benefit of that mutation without actually having that mutation. So this is the type of drug that is going to be very common in the future. And we call this kind of drug an informational drug. So traditional small molecule drugs are chemical entities that deliver where they deliver and have activities that are all determined by their chemical structure. In other words, it's hard to separate what we call the two properties of a drug where it delivers and what it targets. Where it delivers is determined by the dianophore. What it targets is determined by the pharmacophore. For a traditional chemical entity, those are difficult, if not impossible, to separate. For an informational drug, these two properties, the dianophore, where it goes, and what it targets, are easily separated. What it targets is determined by programming. So you know already when the mRNA vaccines are updated, what are they doing? They're typing a new sequence into the synthesizer that makes the RNA. They're basically changing the sequence. That's all they have to do. They don't have to do anything to the packaging. The nanoparticles are the same, but the sequence is altered. So you can easily alter the pharmacophore, or what gene is targeted. The same is true for these drugs that work in the liver. So the PCSK9 drug is programmed with this information, and where that drug goes is determined, in the case of the liver, by putting the ligand GalNAc onto the end of this drug. So this is what these molecules look like. They're bigger than this, and they have added to one end, you've got the RNA, they have added to one end a ligand, or a binding site, if you will, that will determine where those drugs go. This molecule was made by Anastasia, and my colleague John and Anastasia are two of the chemists that we hired at UMass. This one is just two of these molecules joined together with a short linker. This molecule delivers beautifully throughout the brain. It gets into the deep parts of the brain, it gets into the glia, the astrocytes, everywhere. It requires an infusion into the CSF by lumbar puncture or by intracranial delivery. And we expect the drugs are in clinical development through this company called Atalanta, which we formed in 2017. Atalanta is funded and has large partnerships with biopharma, including Roche and Biogen. Atalanta is targeting ALS, Alzheimer's, Parkinson's disease, Huntington disease, lots of different diseases. It can target essentially any disease where the causative gene is known and where there's a likely benefit for lowering the levels of that gene. These types of drugs typically lower the activity of their target. Comanche Biopharma, we started just 2 years ago. This company is focused on one target, which is the causative gene for preeclampsia, which causes preterm delivery because the mother will die if she doesn't deliver the placenta. And this is a leading cause of mortality in the third world, where C-sections are not readily available. In the first world, it's a major heartbreak for many women because they have to choose between their own life and the life of their infant child, who may be close to term but not quite ready. And so this is when you hear the mother's on bed rest, et cetera. It's a very, very serious condition of pregnancy, and also a very common one. I'm sure there are many cases here in the island. But the exciting thing with this drug is a single dose subcutaneous should lower blood pressure within 3 days based on the animal studies, and should enable the mother to carry the baby to term. Let's hope it works. It's going into women probably next year. And we hired the same clinical development team as developed Leqvio. We got exactly the same team because Novartis bought Leqvio for $9 billion, one drug for $9 billion from the medicines company, and we got the clinical team from the medicines company, who were now out of work, to take on this project, which we had been having such a hard time developing because nobody wants to inject anything into a pregnant woman. But luckily, they had seen at the medicines company, they had seen that these molecules were incredibly safe. And so they weren't worried about it. And so we got this great clinical team, and I believe this drug will work, and I believe it'll be in women probably middle of next year, and possibly definitive results could be coming within a few months after that, because the activity should be very rapid. As I said, just 3 days. The clinical trial should be very straightforward. And this company we're launching now, called Diana Therapeutics, because we can deliver to the muscle, the lung, fat, kidney, heart, ocular. We can also deliver into the skin. We can get good activity with transdermal injection, and you can think of lots of applications for elective applications for pretty much for all of these. We have a mouse, and I don't know how well it will translate, but a mouse model for muscle delivery using the target myostatin. This is a target that is inactivated in racehorses. When you inactivate this in a mouse, the animal increases its muscle mass by 30% in 30 days. I mean, I don't know if that would work, but I know I would love to be able to increase my muscle mass by 30% without going to the gym. But there are actually a lot of applications, for example, in balding and in improving collagen, et cetera, in skin, dermal delivery, also pigmentation in the skin is a big medical target. There are tons of targets in the skin that will probably need to be developed eventually. I wouldn't say they're the most high unmet need, because a lot of them are, more like cosmetic, but there's a lot of opportunities there. I was thinking during the earlier panel that having expertise in delivering this type of therapy could be something, if you could do it at a good cost, lower cost would be attractive, because people could come have a few infusions or injections or whatever once a year and have nice, beautiful skin or better hair growth or hopefully better heart condition. Then maybe you could have treatment center where you get all of your boosters, if you will, all at once, because it's going to be like vaccine. And I think it's not a cure; it's a therapy. But you're going to need to do this over and over again. I already mentioned that. One of the things that's so cool is when cells take up these search queries, they don't all go in to the search engine at the same time. That might be overwhelming. They end up in these intracellular vesicles shown here in green. The drug is in red. So when you give this drug, it ends up getting into the cell. This is a cell. The nucleus is here. And you can see these green particles. These are endocytic vesicles. They're actually the digestive system of the cell. They're called the lysosome. And this is why early versions of the drug didn't work, because the drug's going into like an acidic, environment where the RNA would be normally destroyed. But because these molecules are now stabilized, they survive basically inside the lysosome of the cell, and they get released slowly over a period of months, and that enables these drugs to have a very long, effective period, and hopefully will make it more useful for treatments, for example, for infusion into the CSF, although that's a fairly invasive procedure. If it's only once a year, that's not so bad, especially when you compare that to the devastation of a neurodegenerative disease like ALS, where you basically are going to die if you don't get a treatment. So I'll just end here with a picture of my daughter at the Nobel Prize headquarters, and she's gathering medals. And when we went there, I told her, "Vicki, grab as many of those as you can." And so she was doing that. But it's actually a very poignant image in a way, because these are special gold medals. They're filled with chocolate. And Vicki has type 1 diabetes. So, I got inspired to get into
My line of work, because of the cloning of the insulin gene into bacteria, and right here in this picture, under her sweater, she has an insulin pump that's delivering insulin to her every day, and that's why she's alive, because she developed type 1 diabetes at the age of 1.5, and her body lost the ability to make insulin for itself. So now her insulin is made for her by bacteria and put into a pump, and she gets insulin every day. And for her to eat one of these gold medals, she has to give herself insulin. It's a therapy that keeps her alive. Without it, she would have died by the age of 2, probably. And actually, I should say that my grandparents from Maya, I think one of the reasons they left Maya, they thought the water was bad. The story is that they lost like 3 children in their childhood before they emigrated, and they lost more in the United States. I wonder if we had type 1 diabetes in our family. We have autoimmunity in our family because a lot of us have various issues with psoriasis, my other siblings, or Hashimoto's disease, which is an anyway, autoimmunity is a big problem. It drives type 1 diabetes. It may have driven my family to leave Maya. And then it inspired me to work on something that hopefully will make new therapies like this, medicines that they don't cure the disease, but they basically allow you to have a normal life because they correct the underlying problem. In this case, by giving the bacteria the genetic information to make human insulin. In my case, with RNAi drugs, we can give an RNA that enables a patient to inactivate a gene that's causing disease, or a gene whose inactivation will make you even healthier than a normal person would be, like the PCSK9. And so I think there's an incredibly bright future for this type of medicine. I think it will be the major type of new medicine because it's so much faster to make them. And you've already seen it with the mRNA vaccine. It took about 3-4 months to make the mRNA vaccine. This is the same with this type of drug. And cost of goods is actually quite low. So the main bottleneck is testing. The clinical trials are incredibly expensive. But I think the future is extremely bright. And I'm very excited by, learning more from cells about the tricks they have, because they certainly have many left. And I'll stop there. I'd love to entertain any questions that you have. Thank you. Any questions? Sometimes it takes a little while, I found out, before people get up the nerve to ask a question. There's no bad questions. It's one thing I learned at the dinner table. My mom and dad had, because they're so different, paleontologist and an artist, they were very good at having a conversation. And you learn a lot that way. But it's been a long day. Maybe people are tired. Okay, well, thank you.
Temos que estar mais à frente. Muito bem, interessante.
Muito obrigada. Muito obrigada à doutora Luísa Mota Vieira e muito obrigada ao professor Craig Mel. Thank you, Professor, for your amazing presentation. It was really, really, really an honor to hear you and to meet you in person. Thank you very much for coming to our summit.
Thank you.
aproveito desde já então para dar seguimento ao nosso Summit, apresentando a doutora Cristina Calisto, Presidente da Câmara Municipal da Lagoa, que irá proceder à sessão de encerramento.
Saúde o Sr. Presidente do Governo Regional, aqui presente entre nós, sou o Dr. Luís Farinha, Presidente do Conselho de Administração do HIA, digníssimos convidados, participantes, oradores deste primeiro Summit Internacional promovido pelo HIA, profissionais de saúde aqui presentes, pois nestes 2 dias, neste encontro, muito se ouviu, muito se disse, muito se debateu à volta da saúde, das técnicas, da importância que o HIA tem no contexto regional, o seu papel na investigação e na inovação e a sua importância na formação. Foi para isso que o HIA concretizou este encontro, debater os desafios, olhar para o futuro e perceber aonde o HIA tem lugar nos Açores. Não é por ser na Lagoa, mas por tudo aquilo que eu ouvi, o HIA é necessário. Responde aos utentes, complementa os nossos serviços de saúde, vive em paralelo com outras entidades privadas na saúde e até à data o que revelou é que há espaço para todos. A nossa saúde é a maior das prioridades. Precisamos que Portugal e os Açores também sejam referências na área da saúde, num tempo que se exige respostas cada vez mais científicas, humanizadas, imediatas e determinadas. As pessoas mudaram, os tempos também mudaram e esta mudança chama-se progresso. E é neste progresso que temos que estar focados porque não podemos, isto implica não estagnar. Por isso, caros convidados, congratulo-me com a existência do HIA pela posição também de notoriedade que traz a este Conselho. Esta semana ficamos a saber da intenção de compra do HIA pela CUF e aproveito esta ocasião para dizer que é mais um passo nesta caminhada de exigência que o utente quer. E vejo a chegada deste grupo económico como uma situação positiva na área da saúde e que na condição de insularidade que temos ainda encontro mais vantagens. Desviando-me da saúde, mas falando de um aspecto que aqui foi focado neste encontro, abordando em específico a questão da formação, ontem partilhei algumas ideias com o Sr. Presidente do Governo Regional. A necessidade de olharmos para aquilo que são as efetivas preocupações do nosso mercado empregador da atualidade. E neste espaço do Tecnoparque, situado na Lagoa, é tempo de pensar num centro de formação e capacitação de recursos humanos. Se a Universidade, como ontem foi referido, pode ser um grande parceiro na formação na área da saúde, já na área das novas tecnologias, da engenharia informática, o Nonagon pode ser o parceiro estratégico. Atualmente não há nenhum engenheiro informático desempregado nos Açores. E isto quer dizer algo, significa que neste momento em que muitas empresas internacionais olham para nós com interesse, nós não temos tempo a perder, porque ficar aqui implicará ter os recursos humanos necessários. E se levarmos muito tempo a encontrarmos as soluções, vamos perder também esta oportunidade. E este Summit veio reforçar essa consciência de urgência na capacitação da nossa mão de obra cada vez mais qualificada. Fica aqui por isso o meu desafio para, conjuntamente com o Governo Regional dos Açores, podermos encontrar as soluções adequadas a esta nova necessidade que se apresenta perante os nossos tempos. Mas voltando ao tema principal que nos reuniu nestes 2 dias, estou convicta que este Summit foi mobilizador de vontades e felicito por isso a equipa do HIA, todos os seus colaboradores, os seus responsáveis e em particular o Dr. Luís Farinha, pelo seu sentido de compromisso, pelo seu sentido de servir e por ajudar todos os açorianos e não só, também aqueles que por aqui param e que nos procuram. a nossa saúde merece. Uma palavra também de referência a Greg Mell, aqui presente, que muito nos honra com a sua presença neste evento e que será distinguido justamente pelo Hospital Internacional dos Açores, à qual eu também terei a oportunidade de, simbolicamente, lhe entregar a medalha de mérito municipal, a medalha de mérito eu peço desculpa, não é a medalha de mérito, é a medalha comemorativa dos 500 anos da Lagoa. E isto porquê? Agora fiz aqui um raciocínio muito rápido. Ontem estive numa conferência, depois desta, sobre cultura e sobre Gaspar Frutuoso, e percebi que 1522 foi um ano muito importante. Pelos vistos, faz 500 anos das Romarias em São Miguel, faz 500 anos do nascimento de Gaspar Frutuoso, estamos a comemorar os 500 anos da erupção vulcânica de Vila Franca do Campo. Portanto, 1522 foi seguramente uma data muito importante. E também foi há 500 anos que a Lagoa passou a ser uma vila e um município. E espero que tenha sido por bons motivos, não seja por causa do vulcão, tão negativo quanto o vulcão. E Gaspar Frutuoso, na sua obra Saudades da Terra, faz uma distinção entre o antes de 1522 e o depois de 1522. E caracteriza as nossas ilhas antes de 1522 como algo idílico, quase paradisíaco, tudo era maravilhoso, tudo era tão bom. E depois de 1522 parece que se deu o apocalipse, tudo correu mal, tudo parecia ter se tornado nebuloso. Mas hoje estamos em pleno século XXI, os tempos são tão diferentes, acho que temos tempos complicados, provocados por um contexto internacional que marca também a nossa vida diária, mas creio que os Açores estão a percorrer um caminho muito importante nesta fase, que nos estamos a tornar também notáveis no exterior e temos seguramente muito para oferecer para quem aqui nos visita ou escolhe a nossa terra para viver. Por isso, eu espero que esta oportunidade também seja uma forma de o convidar, Greg Mell, a visitar a Lagoa, a conhecer um pouco mais da Lagoa e a saber também da nossa história. A ele e a todos aqueles que nos visitam também, o meu obrigada. A todos vós, o resto do bom fim de semana e obrigada pelo convite. Muito obrigada.
Bem, como sempre ouvimos dizer, tudo o que é bom acaba depressa. Ai.
Youngest city of Portugal, 10 years from city, 500 years of village. I hope you enjoy. Thank you.
Muito bem.
Como eu estava a dizer, estava aqui a comentar com o Alexandre, quando estávamos ali sentados, de facto tudo o que é bom acaba depressa e realmente foram 2 dias que passaram muito rápido. Obviamente que foram muitos dias até aqui chegar, mas depois passaram muito, muito rápido e foram fantásticos. O nosso agradecimento, o meu especial, porque eu não sabia estar aqui, isto foi uma novidade, mas efetivamente senti-me bem a fazê-lo, porque efetivamente é uma honra estar com todos vós e fazer parte daquilo que é a família do HIA, que tem sido para mim também durante mais de um ano e, portanto, desde já agradeço a todos, aos meus colegas, às pessoas todas que estão cá. Obrigada por nos terem ouvido.
Muito bem. Muito obrigado a todos mais uma vez e, portanto, seguimos agora para as instalações do Hospital Internacional dos Açores. Não sei se querem acrescentar mais alguma. Bem, seguimos então para as nossas instalações, onde se vai seguir a sessão de homenagem ao Sr. Professor Craig Mell das nossas instalações. Muito obrigado e o resto do bom fim de semana.